Paul Berg, creation of the first recombinant DNA molecule. Recombinant DNA technology is an evolutionary advancement. Recombinant DNA is the joining together of two or more DNA molecules from different organisms. The first recombinant DNA was created by Paul Berg, an American biochemist and professor emeritus at Stanford University. Paul Berg is known as the father of genetic engineering. In 1953, Watson and Crick discovered the structure of DNA, which led to many discoveries such as correct interpretation of the genetic code and central dogma of biology. The discovery of bacterial transduction by Norton and Zinder led Paul Berg to think about introducing new genes in the existing DNA. Paul Berg was trying to develop a transduction system for mammalian cells to facilitate genetic modification, similar to bacterial transduction system, where bacteriophage P1 infects E. coli but comes out as P1 particle containing DNA segments of E. coli genome. So are genes picked up by viruses from one mammalian cell and transferred to the next host cell detectable? Mammalian cells have a large genome compared to a virus, so the virus cannot take up mammalian genomic segments. The catenation of DNA molecules led Berg to think if he can join two DNA molecules together and put that molecule in the host cell. Paul Berg took DNA of Simon virus SV40, whose common host is a monkey and is known to cause cancer. He also took lambda divigal plasmid containing lambda-phage DNA and gal operon. Both the plasmid and SV40 genomes are circular, so they were cut open by the restriction enzyme EcoR1. EcoR1 cuts circular DNA once, creating linear DNA. Then, to prevent rejoining of cohesive ends, he used 5' exonuclease to cleave 5' ends on both SV40 and lambda divigal to synthesize poly-A tail at 3' ends of SV40 and poly-T tail at 3' ends of lambda divigal using known enzyme terminal transferase. Such complementary extended ends are called sticky ends. Then, SV40 and lambda divigal with added sticky ends are mixed together, annealing condition form non-covalently associated DNA molecules. Since A and T sticky ends were only approximately similar in size, there were gaps at A and T junction, which were filled in by DNA polymerase I and sealed by DNA ligase, creating the first recombinant DNA molecule in 1972. The result was that he was able to create a recombinant DNA molecule, but the actual experiment was not able to finish. The idea was to propagate recombinant DNA molecule created in E. coli and check for inserted gene expression. However, Berg did not insert SV40 lambda divigal hybrid DNA in E. coli since he realized that adding tumor-causing SV40 in such a universally common bacteria would be equivalent to planting a ticking cancer time bomb in humans, who might subsequently become infected by altered bacteria that escaped from the lab. So rather than continuing this groundbreaking experiment, Berg halted his work at this point with concern that recombinant DNA may lead researchers to perform extremely dangerous experiments. Shortly after, Berg and colleagues called an Alzheimer's conference with scientists around the world. Berg drew up professional standards to govern research in the new technology and published in 1976. Since SV40 was tumor-causing, Berg was not able to insert the recombinant molecule he produced in E. coli, and no conclusion was made regarding exogenous gene expression in the host cell. Over time, many regulations have been aced. Scientists discovered transferred DNA can replicate and can be used to produce proteins needed, except recombinant organisms that include extensive DNA regions from pathogenic organisms. Paul Berg won a Nobel Prize in Chemistry for his fundamental studies of biochemistry of nucleic acids, with particular regards for recombinant DNA in 1980. The recombinant DNA molecule technique found by Paul Berg allowed Cohen and Boyer to create the first recombinant organism and introduce concepts of transformations. Recombinant DNA gave new methods of diagnosis, prevention, and treatment of numerous human diseases, such as diabetes, by insulin. Today, recombinant DNA is used in therapeutic products such as vaccines, growth hormones, antibodies, recombinant proteins, anti-cancer drugs, as genetically modified crops, microbes, and animals, in diagnosis as gene therapy, gene mapping, CRISPR, and distinctly in energy processes such as biohydrogen. Thank you for watching.